Okay, so today is the last lecture, the 23rd lecture, and we're going to discuss issues surrounding reproducibility in life science research, a topic that we've touched on a number of times in our discussions throughout the semester, but I'll try to bring some of these concepts together. Well, we certainly know this figure by this point in the course. We've looked at the bioinformatics that are upstream, where the databases and data portals that we grab data from, and we've certainly looked at the computational biology and modeling in different contexts, and we've looked at the tidyverse that facilitates the visualization and exploration, data cleaning, the data wrangling to get your data from those bioinformatics into that Tibble, into those convenient data structures. But today I want to talk a little bit about communication and where things can go wrong, and how modern approaches offer solutions, or at least mitigate the issues to some extent. In a way, we're back at the beginning of the course. You may not realize that, but you might recall that we had discussions about systems biology, about moving biology in a direction where data is generated in high throughput manners, or more importantly, mechanically, like a pipeline or a factory. And we had discussions about, conceptually at some level, what is the difference between systems commerce like IKEA or a chain, information systems, and systems biology, right? These concepts, what do they have in common? And really, what are the advantages that these ideas have? So what are the advantages of chain restaurants? What are the advantages of chain furniture stores? What are the advantages of well-designed information systems, for example? And what does biology stand to gain or lose from that approach? And we had discussions before where there was a lot of very valid hesitations about moving biology in those fields, in that direction, for lots of different reasons. But let's revisit some of those issues today. Some of you may have heard of the reproducibility crisis. The wiki page in the reading gives a pretty decent overview of the main problems. The fundamental observation is that many studies in the literature are not reproducible by third parties. Now, here the studies, for us, are mostly related to life sciences, but the reproducibility crisis extends to other areas, economics, for example, both medical and basic research communities in biology and the life sciences. But computer science and many other areas are affected by this. Now, there's some disagreement about the severity of the problem and also how to deal with the problem. Firstly, I would tend to agree somewhat that it's not as severe as perhaps what some people present it to be. I'm not sure, it's gotten any worse over the last 2 decades. I think that there's maybe more science done, and science moves at a faster pace. But in my opinion, and it's just my opinion, it's probably always been the case that science has been difficult to reproduce. Now, I think that because we move faster, because there's more people doing science, I think it's maybe perhaps more brought into the light. On the other hand, I think that there are novel aspects of science that make reproducibility more difficult. And I think, you could reflect on that if you stop the video for a second and ask yourself what those issues might be. But to me, for example, genomics causes a problem. When you when you pay quite a bit of money to sequence, say, 1,000 samples from a clinical cohort of, let's say, cancer patients, and you submit that paper for review, it's certainly not possible for a reviewer to drop everything they're doing and sequence those same samples in a reasonable amount of time and cost and everything else. It's just not at all feasible. It's obvious, right? So how can a reviewer or a third party in general, after the paper's been published, have any hope of reproducing that study. I mean, the material itself has been used, so you can't sequence the same patient twice, for example, and it's very expensive projects to begin with. And well, there's other issues that come up, but definitely that the timeline, the time scale that we review papers on, which is usually on the order of weeks in life sciences, negates the possibility of very close analysis of the data. So even if the people were to give you the data and you say, okay, well, I trust that it's been generated properly, I trust that they've handled it well, I trust that there's been no dishonesty, for example, it's still difficult for the reviewer to find the time in a couple of weeks to redo all the analytic analysis, especially if they don't have the code and computational infrastructure, or if proprietary software is used. How do they need to pay for the license themselves of that software that the original authors used? There's many issues that get in the way of reproducibility, and there's disagreement as to how to deal with the problem. So if studies are not reproducible, some people are suggesting that, well, it's kind of good for science in a way, that people are on the edge, right? That essentially, it's an Australian, I can't remember his name right now. I tried to find his tweets from Twitter, but I honestly, I couldn't. But if I could paraphrase his argument, it was basically, I believe, that we shouldn't get too dogmatic about making all of our work reproducible. We want to be creative in the lab, right? And physicists are creative, for example, with string theory. They come up with theories that take maybe decades to then validate experimentally, just because there's no way to validate them until a technology comes along that makes it even close to feasible. So do we really want to be too pedantic to say that only things that are absolutely proven to be true make it to the literature? Maybe even if it's not quite true, the ideas there and the forward thinking have value to the community. And I think there's some value to that. We don't want to be too dogmatic as scientists. We want to maintain our ability to be lightweight and to explore. But if really there's a significant fraction of our papers that we're putting in the literature that have flaws in them, and what can we do about it, right? And I think that the field of quantitative life scientists, that's whether you're a data scientist or a bioinformatician or a computational biologist, this field as a whole has a tremendous amount to offer to addressing this issue. Because, to go back to this example, by well-designed information systems, they're going to support good biology, right? They're going to make it more accountable, essentially. Okay. Well, okay, I guess what I don't mean here with the reproducibility crisis is absolute, obvious, complete dishonesty, and there's clear examples of that. Now, my opinion, it's just my opinion, is that research community is actually incredibly efficient self-policers. We don't have tremendous numbers of scandals with the fabrication of data or the misanalysis of data intentionally. Now, there are examples, that's for sure. In the reading, I'll give you two. I gave you two. One is the Duke scandal from Duke University back in the early 2000s, and the second is around the company Theranos. And there's an excellent book, and there's a movie on Netflix about Elizabeth Holmes, and I highly recommend it. Elizabeth Holmes and Theranos is more about biotechnology, where the Duke scandal is really more about bioinformatics, cancer informatics, and genomics. Now, certainly we need to hold our community accountable, the members of our community accountable for outright dishonesty. I don't think that this should be, there's other issues of reproducibility that are more insidious. Even when somebody's being honest, or they can be careless, right? And that's more, I think, where the reproducibility crisis is seen. So I do recommend, I did put some links to the reading of the Duke scandal. Let me just say a couple words about it here. Basically, a very high level view of this is there was a postdoc, Neil Potty, at Duke University, in a very famous breast cancer genomics lab. And they had extremely good collaborators in biostatistics who, that it's there was no lack of skill. But the postdoc, Anil Patti, essentially he was working on a problem related to breast cancer using genomics. They were profiling breast tumors like we've seen now many times in the course. And he came up with a signature or a set of genes like Oncotype DX from a couple classes ago, or Mammaprint, right? And it was basically to predict patient benefit to a certain chemotherapy. Now, to make a long story short, that lab moved, and the associated clinicians at the hospitals related to Duke moved very quickly to take that signature, that Oncotype DX-like entity, and bring it into the clinic in prospective trials, I believe. So the point there is that these patients now were, their tumors were being profiled with the Duke signature, and that was being used in the context of a trial to determine patient therapy, right? So now, the paper had been published, I believe, in PNAS, if I recall correctly, Proceedings of the National Academy of Sciences, and a couple really, really good biostatisticians at MD Anderson, I believe. I think they were both at MD Anderson. They started to add questions about Anil Potty's signature, and they tried to reproduce the study themselves. And I think the data was available. There was from the publication, which was good because the PNAS forces, the journal forces the authors to release the data. So that's a positive aspect that this could be caught. And when they started to look at and try to redo what Anil Potty did, they discovered that basically some of the genes that were in Anil's signature weren't even on the microarray at the time. I believe it was an Agilent array. And well, that's hard to understand how genes that aren't even measured by the microarray end up being in the signature of benefit to this chemotherapy. And they found other errors that looked perhaps careless, but some seemed to be just clearly hard to understand how everybody could make them. And they started to raise the alarm, and it took a little while, I believe, but eventually the community at large did see that this was the signature had been faked, that Anil Potty had made up this signature and it wasn't reproducible in the data. And now the tragedy there is that women were actually being, the treatment for some women at the hospital there were being determined by a fake signature. And this was big news. And Anil Potty was immediately released from North Carolina. There's a long story about the PhD student that was working underneath the postdoc. He was a PhD med student whose career was incredibly damaged by the situation. He tried to stand up and to go to the PI, I believe, even to the dean of the university, and because he had caught sort of a sense that the data was being faked before publication, but he was ignored. And I think, the leader of the group, probably as a PI, I can sympathize that you have a good postdoc, we think is a good postdoc, and they're plowing forward, and they're getting results. And it's difficult, even when you're the supervisor of a PI, to validate the findings firsthand. Take, for example, I ask you to put yourself in a position right now where you were to go back to your supervisor, or whether it's an academic group or the company that you join, and you say, hey, I took this just incredibly amazing course from an even more amazing professor, really just a super awesome guy, and I learned so much, and I totally think that we could profile these tumors, and I could do all the data science for it, right, et cetera, and you'd go back to saying how great the professor was, etc. And then, you know, your company says, or your group says, yeah, sure, you know, like, that's great, go for it, you know, you do it. I don't know anything about that stuff, right? I can't, you know, I don't know the statistics for building a linear model and looking at interactions, etc. But, you know, I'll trust you, right? Well, you go away and you know, you do all those experiments, etc., and you get to the data science part, and, you know, you get a little bit confused because of your TA's notes, etc., but you struggle through it, and, you know, and then, you know, the company wants to market it, or the academic group wants to publish it, and who in the group can evaluate it, right? I mean, they don't have the skills to reproduce what you're doing. They can take a look at it and say if they think it looks reasonable or not, but they can't really reproduce it because your supervisor just won't have the informatics skill. They won't have interacted with this completely amazing professor in that awesome course, which you guys have, and that's something that's, you know, really positive for you. But they're stuck, right? They have to trust you, and well, that's clearly going to be problematic, right? If what you've done has a mistake in it, never mind complete dishonesty. Well, Anil Patil was released from Duke, and I think that many people were reprimanded. Let me also say that the biostatistician who was a co-author on that paper, he certainly had the skill to evaluate what Anil had done. I think, though, I know that professor, he's an excellent biostatistician. I think that it's difficult for any one person to really get in there and validate, you know, even if you have the expertise, right, to find the time to go through and check every aspect of your postdoc's work. At some point, you need to trust the people around you, and when they set out to be dishonest, you know, it's very hard, but certainly it's something that I think a lot of PIs and people are worried about. And so, well, at the end of the Duke scandal, which really took a long time to resolve, and there was lawsuits from the women who were treated potentially incorrectly, and it was really took a long time to resolve. I think 2016 there were still cases. Well, yeah. So at the end of that, there was a call to write down specific rules of how bioinformaticians should behave and how these kinds of studies should be done. And one of them, of course, is that you shouldn't move a signature that has only shown up in an academic paper that quickly towards the clinic without a lot of extra checks and balances. What are the causes then of irreproducibility outside of these egregious, dishonesties? Well, simple clerical mistakes can happen for sure. That can make it very hard to reproduce somebody's paper. I'm not sure how we can really fix that, except I think you trust some labs more than you trust other labs because they're careful and they have their ways of checking things over, right? Multiple people writing on papers, etc., that are really intellectually involved in a project, maybe not just as collaborators for specific aspects, but really understand the completeness of the paper. All aspects of the paper is what I mean. Poor or incomplete description of the results, certainly, right? There can be just a misunderstanding of what that result means. It can also be that the method is not described well, and that's really a big problem. And I think the journals have moved towards some solutions in that way. I think things have changed. I know as a bioinformatician, it used to be very difficult to submit highly technical mathematical or statistical analysis to some of the journals. They were pushing it for it to be shortened and abbreviated, and that's not necessarily good for the mathematics, right? I mean, mathematics takes what it takes to describe what you're doing, and we can push that the description is parsimonious, right? That it's minimalistic, but it has to still be complete, otherwise it's meaningless. You can't sort of half do math. And when you're given a priori limits on words and method sections, that can be problematic. Nowadays, I think journals have pretty liberal rules with supplemental methods, etc. But on the more note that I don't think things have changed, and this is just my opinion, is that journals often send biological papers to of course, several authors, and sometimes the statistician or the bioinformatician will get it, and they'll judge primarily the correctness of the methodology from informatics perspective or statistical perspective. And I've witnessed cases where I think the paper has very shoddy methodology, and I've voiced that concern, and it's overridden by authors because the results from the biology perspective look very strong and supportive. I think that we have to be careful that if those results are strong at the biological side, there has to be a way then to still correct the description of the methodology or the actual methods used. This paper should never still be accepted based on what the quality, the excitement of the story rather than the technical correctness. There's other reasons that are insidious for reproducibility, and they have nothing to do with carelessness, perhaps, and nor dishonesty in any way. And many times it's because of technical or selection bias in the cohort that you're studying. For example, at McGill, when we profiled breast cancer patients, they were coming from the Royal Vic Hospital. Now, the Royal Vic Hospital is not your average hospital in Quebec. By average, I mean it's just that it's a research hospital. So the tough cases of breast cancer in the province, many of them go to the Royal Vic. This includes rural areas of the province. So the tumors that are sitting in the freezer there already have some sort of selection bias in them. They're not necessarily representative of the population in general. In some ways, you could think they're maybe the most difficult cases, right? And so you're not profiling really a representative cohort for the population at large, right? And we've also discussed, for example, when we talk about BIPOC matters and sampling, why in the selection process you're trying to minimize the variability, right? Minimize the heterogeneity to find strong signals to build on. So, for example, you might restrict your cohort just to Caucasian women, for example, and not investigate or include a small fraction of Asian women or Black women, right? So now that's not because of racism per se, it's because most of the tumors that you have at your disposal are Caucasian, and that's what's going to allow you to get the most statistical power. However, that doesn't mean that what you find, your gene signature or of recurrence or benefit to a specific therapy, is going to work in that population at large. Technical bias is a bit more straightforward. I think it could be that the technology you're using, the mass spectrometry, which we really haven't talked about in the course at all, or the sequencer, of course, or the microscopes even, that you're using for your study may be slightly differently calibrated than what's being used in a clinic or what's being used in some other institution that's trying to validate your finding. And that, of course, there's a concept in the clinical world of clinical validity, which is different than the concept of clinical utility and technical validity. So, basically, you might have an assay that has promising clinical utility. For example, being able to find the patients that won't benefit from chemotherapy has great social economic benefits. But if it's not technically valid, if your signature that you built in your lab doesn't generalize to the clinic, to that population, it doesn't have technical or clinical validity, right? Okay. Now, as machine learning experts that you are, you also have the understanding that if you're not given access to supporting data, and that includes the training set of both positive and negative examples, you're going to have a hard time to reproduce somebody's result. And of course, there's lots and lots and lots and lots of examples of people who misuse the training and testing and validation data sets. So, they train and they find a classifier, like a logistic regression or regression that we've seen in class, and then or a convolution neural network like we've seen, or they find using an unsupervised technique like k-means, they find subtypes, and then they evaluate the accuracy of that classifier or scheme using the same data or part of the same data. Sometimes there's really small insidious problems in the way that the training and validation data sets are set up. Maybe the data sets are all normalized together, and that actually carries information between training and validation, etc. It can be kind of tricky. It seems like it's pretty simple. It would almost, but it actually can be a bit tricky that there's no information flow from the training to the validation. And really, for example, from the Duke scandal, one of the findings or recommendations was that the classifier that a group like a bioinformatics group finds should be written down in stone. By that I mean you declare it to the public. You say, these are my genes, this is how I combine the genes, and this is my endpoint that I'm studying. Okay, so for example, my genes are the PAM50 genes, my endpoint is prognosis, and my algorithm for combining that data is a regression, let's say. Okay, so now in a validation study, I shouldn't be the one that does it. I should pass it off to a group, a biostatistics group, for example, that evaluates my signature independently of me. I have nothing to do with it. That's very close, actually, to how pharmaceuticals and academic groups get treated when they're trying to put a drug out there, or when they're trying to establish a biomarker. At some point, you need to pass it to other people for independent validation. Other problems is that, for example, as you guys now fully understand, is that you can have bugs in your code, right? There's no, you guys should understand fully that even small programs can be difficult to write, and they can contain subtle errors that you won't notice, and that will just influence your results. And if you don't make that computer code available to the public, then of course there's no way to find those bugs, right? And your study will be irreproducible. Now, how do you avoid bugs in computer code? That's a great question. That's a really hard question too. You have to assume there's always another bug. Now, there are formal areas of computer science that software engineering is sort of a field between computer science and engineering that studies how large software systems are made. And by large, I mean these systems might have millions of lines of code. In fact, the driverless cars have very large computer systems. Lengths of the code is very large, I mean. So, the code that you've been writing in this course is, I think in total for the whole course, is about the amount of code that would go into your average iPhone app, let's say. About 1,000 lines of code is probably what you've written in this course. And that's probably pretty comparable what's in your average app on an iPhone. When you look at the most complicated things we have out there, driverless cars, Google, Facebook, etc., they have on the order of millions of lines of code. So you have to assume that there is bugs in there. Our operating systems too have very large code bases. Lack of statistical power in the study. So we didn't talk about power in this course. That's a really important concept for biology. I hope that you have had training in power analysis from the biostats courses or elsewhere. So basically, if you don't have enough samples to study, and you have a finding, it may be a false discovery. It may just be by luck that you discover a pattern that's very consistent between your data and very consistent in your data, and say aligns well with patient recurrence or benefit to therapy. But if you only have 100 patients, say, that might when you look at the 101st patient, it may not hold, it may not generalize, right? That's related to issues like the improper use of training and validation data set. But it sometimes but it has aspects that are unique. okay, that aren't captured by just the way that you created your training and validation data set and looked at its accuracy. It also has to do with other issues about your initial cohort. And there's not much you can do about that. I mean, if you could only get a certain number of tumors to study, then you're stuck with that. But, well, which is okay, because small studies are often really informative, right? They can pull out interesting data and hypotheses, but you can't state them as fact. And of course, you don't know that yet, right? You need larger studies to really investigate those issues in a meaningful way. And I think that's the, what happens now is a little bit more in that direction in the community, where we're aware that our studies are limited, right? And they require further effort before they're used, say, in clinical practice. Makes sense, I think. There are also improper or naive use of statistics or interpretations of p-values. P-values, for example, in this course, we don't, again, I took the focus away in the course from statistics because of the varying backgrounds here. But if there's one thing that I don't cover enough in the course, it's how important statistics is to quantitative life sciences in all three domains, especially computational biology and data science. Whatever we see, whatever pattern we see, there has to be strong statistical, probability-based evidence for that pattern to be really, truly novel and correct. And, there's a professor from Stanford who's sort of made his career on criticizing many of the biomedical papers out there, including in drug discovery and drug validation trials. Ioannidis has also been famous lately because he's criticized some of the COVID-19 coverage. And, I disagree with him on COVID-19 issues. I understand where he's coming from on his criticisms, and if the link here is to one of his kind of famous short papers about what are some of the causes of the causes of irreproducibility due to improper use of statistics. I won't provide my views here on what he said about COVID-19, but I think that there's a lot of very, very smart statisticians, well-read, very sensitive to these issues, that look at the COVID-19 literature and disagree with them too. Okay. Then there's other issues that we have to acknowledge that contribute to irreproducibility. Society and human nature. We compete, and it doesn't mean that we need to be dishonest, but it does mean that it forces time constraints. We need to move fast. Poverty, we can't generate a big statistical study with a lot of power if we don't have enough money to gather those tumors and to profile them properly and to guarantee enough sequencing depth, etc., enough replicates, etc. And so we're sort of forced to cut corners. And another issue of acknowledgement that I'll come back to very shortly. So I'm sure you have some other things here. So we can talk about this in the lecture of what you think other causes of irreproducibility might be. How can then concretely the quantitative life sciences contribute? Well, bioinformatics mandates include things like the development of ethics guidelines, standards in education. We've talked about that at the beginning of the second part of the course. Data science contributes methods for better expressing our results through visualization and etc. And stuff that we'll talk about in a few minutes, with notebooks, for example. And computational biology continues to improve methodology with integration with solid statistical foundations. In fact, as my training had relatively little statistics in it, and many areas of computer science aren't very sensitive to statistical analysis, and branches of mathematics don't use a lot of statistics. That's something that I've had to pick up over the years on my own. And now, of course, in the training programs for computational biology around the world, there is a lot more statistics from day one. And as a side note here, I think if we could change something at Concordia in our programs, I would recommend more statistics in this direction. Okay, so what about let's look at some concrete ways that we can fix things. The issues like incompleteness in the manuscript. So that could be, for example, poor description of the result or poor description of the methodology. We just talked about that. Well, what about Distill? I don't know if you guys had a chance to look at this link. This is Distill. Okay, and Distill.pub. And it's a new type of journal, right? It's peer-reviewed, can be peer-reviewed, or you can submit commentaries, etc. And it's mostly used, I believe, for machine learning, deep learning, okay, where, now, basically this is all online, so this in case here is like, I guess, some kind of guide to optimizing, something called Bayesian optimization. And this is a published paper, this is something you would put on your CV, but here you can see that the graphics are are worked right into the manuscript with exercises, etc. I mean, certainly there's hyperlinks to the literature, etc. And then you can have things like this, right, that you would not have in, for example, your typical paper style journal, right? So now I can have these kinds of animations, right, that just weren't possible before. And let's see if there's anything else in here. Yeah, so there's a few animations, etc. In other manuscripts, they make use of other types of visualization techniques. So you can see how, for example, yeah, again, see here, this is a small movie, right, that allows you to explore the data. So basically, in these papers, there's a tight integration between the code, the data, and the natural language text. I mean, the natural language text is still there, but it's integrated in with actually the code itself, right? So here you can see they're training a convolution neural network to recognize different numbers, handwriting. We talked about this MNIST data set a little bit. So if I put the cursor on 5, I can see it's trying to learn 5, over remember the term epoch, right? And it gets that right. So if I come down here to 0, you can see down at the end here, whoops, it predicts, it gives its prediction and the truth here. Is there a mistake or something in one of these? I don't know if there's an example of where it gets one wrong. Well, I'll let you play with that. But yeah, there's definitely nice things in there. And if you were to look through all these manuscripts, you would see really nice examples and creative ways of integrating data science into the papers in a more dynamic way. And doesn't this look a lot like an R notebook, right? Okay, so why does this address anything in terms of the issues of reproducibility? Well, I think that Distill is putting the data and code right there for you, right? So if you don't understand the result or the method, you can explore it quite directly, right? And you can look at it actually goes further, right? It also addresses issues of like bugs in the computer code, okay, and the inaccessibility of the supporting data, including the training set. And because you can see that, you can start to assess if they've done the training and validation data set correctly, right? It's all there. It just sits underneath the hood. You can download all of that information right there and then. Okay, so in a similar note, I just wanted to say here is that Distill is available for R, and we're putting our first paper into Distill now. So it'll be published at a different journal, but we're going to have an online version that's much more colorful and dynamic that explores the reef microbiome in Barbados. Hopefully that comes out soon. So I think it's kind of a nice way to publish. Our journals aren't really built for that. They're built for natural language with 4 or 5 static pictures and maybe a video on the website of explaining an overview of the paper or something. But we need something more dynamic, right? Now in that direction, you already know, you've already seen now our notebooks, right? And they're pretty powerful. They're exactly like what you see in this distill. So now, if you make that notebook available, then all that code has to be able to be run, right? It'll be runnable. So the user will have access not only to your paper, your natural language, your interpretation, but he or she will also have access to the code that you ran, the analysis that you chose, how you set up your training and testing set, etc. Everything is right there. And if the person disagrees, they can go and explore alternative hypotheses. This over here is the example in Python Jupyter Notebook. They also have great functionality in that direction. So yeah, I think that definitely the idea of notebooks is a better way to publish, and that's got to be coming, right, through journals and techniques like Distill. Something that we haven't talked about in the course, and it's really important for reproducibility, is Git. And Git is software that guarantees persistent and consistent code within collaborative projects. So what do I mean by that? That's a bit of a mouthful. Yeah, essentially, imagine, right, because you guys are all now coding experts, that you were working in a team of 10 or 15 individuals, or 100, or maybe 1,000 if you're working at Google or Mercedes and trying to design a driverless car. So how do you maintain code in a big system of 1 million lines amongst all the people who are working on it, right? So how do you guarantee that when you change a function that you've written, somebody else doesn't change that same function or give it the same name? How do you, when you fix a bug, how do you update everybody's code, right? Everybody else in the project needs that bug fixed. You're not going to email it to them, right? So what Git does is it's a master, kind of a god that stands over the code in your directory, and there's a there's a repository that's here in green. Okay, so that's the main copy. It's the mother load, right? It's the Death Star, and what you do is that you copy the repository from time to time to your local machine, right? So this is your machine here. And you make changes. That's what these dots are. You have to change 1, change 2, right? Change 3. And then you want the other people to also access those changes. So here, somebody else may have made a change here, and somebody else might have made a change there. And this person that's orange checked out, I guess it's Donald Trump or something, checked out because he has nothing better to do now except screw up around code. So he forks that repository, or clones it, and more precisely, he clones that repository, and he has his copy, and then he makes a change here. Okay, so this is maybe reminiscent of the cloud-based figures that we had near the beginning of the course. So how does then this blue worker get their updates back into the main chain? Is that they now they pulled the repository, now they push it back to the main chain, and Git is the software that integrates those changes back into this green mother load, the Death Star. And now when Donald Trump makes a change over here, when he tries to push the code back, those Git will acknowledge that, well, actually Donald Trump didn't have, because he cloned this repository before blue in this region here, before blue put his changes in. Now Git will make sure that Donald Trump's changes are integrated with the blue workers' changes, etc. So it resolves conflicts, and basically it highlights conflicts back to the user, and there's certain very precise military grade rules that people have to follow for pushing and pulling their work. Okay, so it's used everywhere. I mean, Git now is it's used in my lab constantly. All projects are put under Git, and they're used by the biggest companies out there. Now, there's other competitors to Git, but Git is probably the most famous one. And we actually don't use it in the course. In previous years, I have taught it, but not with the cloud this time for technical reasons. Now, you have a repository and you need some place to store it. And we use in my lab a website called Bitbucket. There's competitors, GitHub and many others. GitHub now is owned by Microsoft. Bitbucket, I believe, is owned by Atlassian. There's minor differences really between them. These are websites that specialize in hosting Git repositories. And so here, for example, this is my Bitbucket page, well, my lab's Bitbucket page. And so each one of these folders here is a repository for a project. So as each student it starts in the lab, they create a repository, and we follow certain rules for describing that repository and organizing it, and they push it to that. Well, part of those rules is to put it underneath Git, so Git controls it, and then we save those repositories at Bitbucket. And that means that any new student starting up, or if I need to take a look at the code, etc., then I can clone that repository easily from the centralized site. So this part's like a cloud, right, where I can grab it at any time. And I can make sure that then I have the most up-to-date version of it. I can make changes to the repository, and I can push them back to Bitbucket, and we'll always be sure, essentially, to have consistent views of where that project is at, because Git enforces those. GitHub is another one. That's probably the most famous Git repository tool out there. We use GitHub too. In fact, this whole course is stored it's right here, data science, right there. That's the all the infrastructure for this course, from the website down to the lectures and assignments, are all stored at GitHub, and I'm going to show you that in more detail in a few minutes. The last issue here. If we go back, back to this. Another issue. One thing we ended up down here was the society and human nature, and why that might contribute to irreproducibility. I wanted to make one comment about that more before leaving. Is, for example, the way that we in journals, the way that we list author order of authors, is really crazy, right? It's from the 1920s or something, and I don't know why we still do it. Now, inside of papers, often we have a section where we can declare what each person's contribution was, and that's good. However, unfortunately, when we're evaluated professionally for tenure, for grants, for all sorts of things, raises, they look for things like how many first authorships do you have as a junior researcher, and how many last authorships you have as a senior researcher. And this is a paper from 2014, but I just want to point out that it's kind of insane that the 15 or so researchers here have to be linearly ordered. And so how much that, even it's completely not true. We all make contributions that were really critical, or many of us made contributions that are really critical, and I'm not sure that one person is more important than the other. For example, I grabbed money for the project and I oversaw it, but two very smart postdocs spent 3 years working on it, and they can be given co-authorship, but they probably spent 50 hours a week working on this for 3 years, and then other students in the lab spent also significant times, right? Other people like here and here, they contributed samples, right, from the clinic. I don't think that their contribution is anywhere less than what our contribution was. It's just different. and other people contributed technical prowess, right? So they are the experts at DNA sequencing, for example. So it's not that we should necessarily have to say that their contribution is less. It's just different. But this kind of idea where we list everybody in a line and we say, okay, well, the first author is really important, the last author is really important, but even second to last or third to the last is somehow less, really causes a lot of problems, in my opinion. I think it's really outdated and it should end, because it really is damaging to many people's career. And I think it's one of these things that facilitate irreproducibility, because because I think people know that if they're not first or last author, they perhaps don't spend as much time on the manuscript ensuring correctness, for example, but other reasons too. Okay, so I'm going to switch now to look a little bit technically at how the course is organized informatically, and hope that you get something from that. Okay, so in the last few minutes of the course, I wanted to show you a little bit about how the course webpage and notes are stored, all using R. And okay, so this is the GitHub, my GitHub homepage, and you can see that I have a few repositories here. So I think I should have mentioned before, one nice thing is that as people complete their projects, when they're working on them, they make them private, and then when you're published to paper, we just flip a switch, and then that code is available to the community. And so, for example, here, there's a repository called mikehallett.tgithub.io, and that is that's the repository for my website. That's this website here that contains all of this information for the website, all the updates and things to do. And you can see it's like a bunch of files, right? And I'm going to show you more, dig into that in the course webpage, which is right down here, Data Science 2020. And you know, there's nothing stopping you. I've made it completely accessible. Is that you come over to it and you can clone it. Essentially, you come into here and you can clone this repository, and it would copy all of the files of the system down to your local hard drive. And if I do that, I can show you. I'm not going to do it now because I have it. This is my personal machine, right? And here are some of my repositories. My path is just into repos, and I can go into Data Science 2020, and if I do an ls, I can see there's a bunch of files there, and so that's a copy. That's a copy of all the code that's in here, and Git allows me it takes care of the copying to and from, the pushing and pulling of files from the main repository, that's this, and to my local machine. And now you can go into each of these files here. So for example, there's a README file at the beginning that just says basically how to get it, and a little bit about what the repository is about. I can explore these guys in here if I wanted to, the files I mean, but at the same time, it's not that convenient to deal with these files directly on GitHub. I just basically use it to as my master repository where I push and pull from. You can see in here, for example, that the course web page is a mixture of HTML, JavaScript, and other things, right? Okay, that aren't important. So basically, if I come into so what you see here is RStudio, not the cloud version, but the version that you can download and install on your machine, and that's how I normally work day to day. So this is running on my laptop at home, and well, I would highly recommend that you download RStudio in R, and then you have it on your machine if you have any analysis needs as you go along in your career. It's free, of course. It's not like the cloud where you have to pay per month. It's free to download. Okay, so I'm going to talk a little bit about how this is structured. So in my file folder down here, which you guys are familiar with, oops, sorry, pretty well, this quadrant down here, I can see that there's a folder called Data Science 2020. And what I can see is that there's a considerable number of files. I'm going to make this a little bit bigger. And this is basically both the website and the course web pages. And so the thing here is that the website, which gives you the overview and syllabus and project descriptions, etc., all those different aspects, right, that you need to have for your course. But it also has the slides, the assignments, quizzes, and solutions. So everything really is contained all in one. There's no separation from the website from the lectures or labs or anything else, and it's all stored in this same directory called Data Science 2020. Now, what I'm going to show you a little bit more later is that there's a window in this upper right quadrant called Git, which you aren't familiar with, and this is basically all these files that are listed here are files that were changed or created as I prepared this lecture, lecture 23. I haven't sent them back to the GitHub web repository yet. They're local in my my directory, and I'll show you in a little bit how I push them, what's called push, back to the main repository. But first things first, I wanted to show you a little bit about how the course is structured. And for example, if I go into content, I can see that there's in turn a lot of folders. For example, if I clicked on let's say assignments.rmd, you'll see that this corresponds, this is the markdown, right, file, which you guys are pretty familiar with right now. Now, if you read the chapter for the chapters for the required reading for this lecture, you'll notice that this is a YAML header that says the title of this document is called assignments, and then something about slugs, which I don't really understand, doesn't matter. And then basically in here you'll see the markdown language that you've probably observed or even used preparing assignments, etc., in our notebooks, right? So if you come down here, it just lists everything. Sometimes you might wonder what's this, these dollar signs, and that's because it's a language called LaTeX that's integrated with the markdown. And the markdown is integrated in turn with HTML. So LaTeX is a classic language that mathematicians use to write equations, well, to write their whole papers, but it's really good for writing equations, etc. And that's how I feel most comfortable modifying text more than with the markdown language itself. So, on the other hand, like here with triple star, that means boldface. And there's other ways, like here I'm making something italics. There's lots of ways to modify the text, and that web page corresponds to this web page, right? Assignments, right here. So you'll see that as you go through here, it's exactly the same text, but you can see there's been italics, and things are in boldface, and it's inserted symbols, etc. And that was all specified by this special language in here that allowed me to ask for typewriter this little code here. What it's doing is putting a Google symbol beside Google Drive, etc. So that's just the assignments RMD. That's how I made that one page. Of course, in the web page, I need one for overview, for example. In the overview, that's the main page for the course. I can go and I can find that file. It's right here, and you'll see that it looks just the same. In fact, I run a little bit of R there at the beginning. So this is just like you see in the notebooks. That's the code to say that the following is some R code, and all I've done here is load two libraries. I'm not sure why. Yeah, there's the YAML header up here that says it's the overview, etc., and some other formatting options. And then if you go down here, you'll see that all of this is markdown code, including links to the NCBI, for example, and that would correspond to, for example, here, see the NCBI is a link, right? So I specify that. Everything is done in the markdown language, right? And the other pages of the website, projects, resources, and the syllabus are the same. So the syllabus is interesting because it in turn is called something else. Let me show you that. If I come back up and I go to data, I'll see this lessons.yaml. That's a YAML file. And this page here is how I specify. You'll notice here it's part 1, lecture 0, lecture 1, etc. And if I go back to the website and I go to syllabus, that code generates all of this nice stuff in here. So basically, this is all an example of what's called blogdown, and that's an RStudio package that RStudio created. And blogdown was designed primarily for blogs, but I repurposed one for this course. In fact, if I go back to the home page, if I come down here, I see at the bottom I nicely cite Dr. Heiss, and I stole his web page from well, that's the guy who I stole it from him, and his course. I don't know if I have a link to his course here, but you can see here it's using his Tuft Hugo scheme from Andrew Heiss, and it uses blogdown from R. I can click on that and show you. Yeah, it's part of the R Markdown suite, and it uses something called Hugo that's basically like R. It's for web pages. I don't know how to explain Hugo perfectly, but it basically something has to render all these files, right, to take your code and turn it into these web pages, and that's Hugo. To be honest, when I designed this course in midsummer, it took about this course page. It took about 3 days to modify somebody else's and make it, more appropriate for our material, right? I should stress that I never used blogdown before, nor Hugo. You just sort of copy and then modify it, and usually it sort of works, right? Obviously, there's times where it's better to have the background knowledge, but you can get by, and you can do really nice things that way, right? So all of that comes back to folders here, then files. That this structure of Hugo knows that it wants information in a certain way. You'll see things like Pandoc, which is a tool that converts MD files into HTML. If you read the chapter 23 to 27. What else do I have to show you? Oh, static. If I go into static and I show you slides, all the lectures are here. In fact, all the images for the lectures I use are in one folder here, right? I just load them into there as I need them. But in static, I go to slides here, right? I can see today's slides. That's this folder right here, and I can make this a little bit bigger to show you. And it has a YAML header and all of that stuff, like the pizza and the marker that I have and the black background. That's all part of the YAML declaration. I just stole that from somebody else, somebody else's code that does all that nice stuff with the chalkboard and notes, etc. And then I can, just like in R notebook, I can interleave R code with each slide. That starts a new slide, and then I can put figures in nicely, etc. And each slide is like that, and I can include links like this. A nice simple notation. I just use square brackets around the text that you want to see. For example, if I hit this knitter button up here and I come in, that just compiles that our markdown code with the YAML, and in a second I should get, this is the lecture notes for this class, right? And that's how I generate those. Where is that? So yeah, the links, like for example, to still here, right? Okay. So yeah, basically everything's all in one spot. And of course now you can go to GitHub, and I've made it publicly available, so anybody else can download my entire course. That includes everything from the slides and overview down to, for example, I'll go back to content here. It also includes, for example, the reading pages. I set those each class, I make them. But also, for example, well, the links to the video, they're on YouTube, and the solutions for quizzes and assignments are all here. You can go in and, where are they? Oops, yeah, they're there. So I just put those in there. So everything's compact in one spot. So this, again, this is called blogdown. It's for blogs. Distill is also from RStudio, Distill for R. That's for academic papers. And there's bookdown for writing books. They're all pretty similar in how they work, and you can master them in no time. Now, what I wanted to show you is that I can, if I made some changes to these files, and that's why they're highlighted here. In total, there's thousands of files now in that system. What's listed here are the files that I've changed or had modified as I created lecture 23. It looks like a lot, but some of those are minor modifications. Like down here, these are figures that I've added for this lecture, right? I put them in there. So what Git noticed is that I inserted, for example, it says static slides images. So if I go down here to static slides images, I put these files very recently in November 30th, and I added that. And so Git acknowledges that I've modified or added or deleted a file in Data Science 20, and it puts it on this list. So now what I need to do is make sure that my whole website recompiles, and I would do that by hitting this button here, Serve Site. And that's going to recompile my website locally. The next thing I need to do is then push all these updates to the master GitHub page. So I'm going to do that now. Do Serve Site. And you'll see it's saying basically it's building the website, and it's finished. And you'll see that the website is locally here. What I can do is I can pop this open here. Right now it's not at the course website, it's just local. That's a local server, but I can test to make sure it worked. I can come over here to the syllabus, and I look down here, lecture 23, and I can go to here, and I can see that in fact, for example, the slides are right there for this lecture, so everything seems to have worked, right? And well, I haven't set the video because I'm still making the video, so right now that's not been updated. It will be updated by the time that you look at this, so it's a bit like Back to the Future or something. And the reading, I can check that the reading is there. Whoops, I see that there's a problem here with the link to the wiki page for the reproducibility crisis, so I should fix that. I'll show you how to do that. I would go back to my page. This is slide 23. I need to find that error. It should be around, let's say, where it was in the readings, right? So I need to find 23 reading, and if I come up here, you can see that there's some nonsense in there. And what I'll do is I'll just modify that text. Leave it like that, and I should be okay. And now what I'll do is I'll rerun, I should save that file, and now I will recompile the website. Oops, once again, after a semester, I forget to do this every time. I need to restart the server, and then I recompile the website. Show that. I can open it up. Now if I come back to my syllabus, lecture 23 reading, I can see that that problem has been resolved. And now I'm ready to push all of these changes to the main the repository at GitHub. And so what I would do is I would come in here and I would commit, what's called commit, all of these files. And I can do that, whoops, I can do that very quickly. And come down here and commit all of those. So basically what I'm doing there is I'm saying I've made these changes, I commit to those changes, and if I hit this commit button locally on my my machine, I'm going to write down here a message saying lecture 23 created. I save it, and now those changes are forever tracked by Git. So that's part of the history. And the nice thing is that I can trace back, if I hit this history button here, and I can trace back to every change that I've made on the course since the very beginning. And in fact, I can not only just see those, I could easily walk back to any of them and reset the whole system so that it was exactly how it looked back on, for example, November 9th. And I could go down all the way down to any of these nodes, and I could walk back to exactly that moment when and get rid of any changes after that date. So it's called rollback, and that allows me to basically, if I don't like some change that I've made, I can kind of reset the system back to some previous moment. And you can see this goes all the way back to, I guess that would be August 13th when I started the website. Okay, so I can go all, I have everything that ever's happened to this repository. And if somebody else started working on this, nobody else uses this except me right now, their changes would also be tracked, and we would know exactly who did what. Okay, now the second thing I need to do is a technical thing, is that I hit that up arrow, meaning I send it back to the cloud. And now what happens is that it's trying to communicate with the repository at GitHub. You see that there. And now if I go back to GitHub, right, I go back to GitHub. Now if I refresh my screen, I should be able to go, for example, into here. I see lecture 23 created, just because there was a commit here. And if I go into static, I can see now in slides that in fact lecture 23 is now stored at GitHub, and that is permanent. Now anybody else in the world could go to this site, go to code, say I want to clone this repository down to their machine, and they would have everything for the course from start to finish, including all the nice examples of code, etc. Okay, hopefully that's understandable. All right, so one final note as we conclude the course. I hope you enjoyed it and that you learned something. I think that you've seen some data science, seen some bioinformatics and some computational biology, and got some perspective on what they're about. I hope also that you just picked up some overall good IT skills, that you're more comfortable with modern computing and understand where it's going, especially things like deep learning and how it might change our lives in the not so distant future, and how it might also, well, certainly will also facilitate research in basic and health life sciences. Okay, so thanks very much for attending the class.